Hi, everybody. Welcome to Love Goes Live. My name is Mark Lefkowitz. I'm a molecular biology professor at St. Michael's College, and I've been making videos on COVID-19 and other sciencey stuff for the St. Mike's community and really for anybody else who might be interested in science. And I thought what I would talk about a little bit today is antibody production and really antibody testing, and go through the three types of tests that are out there on the market and how they work and what they tell you and what they don't tell you, since that's been in the news quite a bit. And as we all know, antibody detection is a big part of solving this crisis. So any further ado, let me share my screen as I always do. Got a nice set of images for you today. Where is it? Slideshow. Okay, so here's how this works. So let's just say that you have an individual. This individual is unlucky and happens to contract COVID-19. If you've seen an earlier Love Goes Live, a lot of this is going to be reviewed because this is really just going over again how antibodies are produced and what they are and what they do as part of your immune system. All right, so within your body, you have a population of cells that are called naive B cells. In many ways, these are like undergraduates, so much potential, and they haven't quite chosen what they want to do yet with their lives. So naive B cells at some point do have to take on a fate, and what they do is when there is a new pathogen inside of your body, a subset of naive B cells will choose their fate, and what they'll do is they'll become plasma cells. It takes about 14 days for them to do this, and what plasma cells do is they produce antibodies. Now, antibodies are these proteins that look like Ys, upside down Ys in this case, and what you want to remember is that this little portion of them here, the bottom portion, is very specific for some sort of surface protein or some sort of surface molecule on the pathogen. So in this case, we have COVID-19. Here's our antibodies. If everything works like it's supposed to, these antibodies will start to paint or cover the pathogen, in this case, the virus. And then what that does is that flags that virus for destruction by our immune system. I think it's very analogous to like when the military is painting a target with lasers for bombing. Antibodies paint a virus, in this case, for attack by your immune system. Okay, the big idea here is that if you have antibodies in your blood that can recognize COVID-19, it is very likely, perhaps very, very likely, that you've had the virus at some point. All right, so that's like the big idea here. And of course, what we're all hoping, and this is yet to be determined, but we're all hoping is that if you have antibodies, that you have immunity for ideally a long period of time, less ideally maybe for a shorter period of time. All right, so there are many types of antibodies. Let me start with that. But there are two types of antibodies that are involved in this initial response, and they are called IgM and IgG. There's actually many types of IgG. And so I want to point out down here, so this is a timeline, right, down on the bottom. And then this is where you have viral exposure for the first time. And then this is going to be relative antibody concentration in blood serum, What I want you to notice is that this is a log scale, right? So this is going up tenfold every increment here. And so over that like 14-day period when those, actually a little longer than that, but during that 14-day period when your naive B cells are becoming plasma cells and start making antibodies, your antibody concentration will go up. And then as you clear the infection and you recover, the antibody concentration will go down, okay? Now, as you can probably imagine, not everyone responds in exactly the same way. So some of us are going to respond maybe a little quicker, and maybe some of us are going to respond a little bit more robust. And then down here in the tail, some of us may tail off longer, and some of us may drop quicker. Okay, so my only point that I want to make there is that when it comes to immunological responses and to antibody responses, we're not going to all have the same response. Okay, let me clear that writing, and I'll point out one other thing here. So let's say then that you want to get antibody tested, and so that's going to happen some point, let's say, in this window here. And so you can see, depending upon the time period at which you are tested, the amount of antibody that you potentially have circulating in your circulatory system, in your blood, in your serum, is going to vary. And the further out you get, the harder it can be to detect potentially, right? Depends on how much is there. But it's going to taper off, and so when you test may affect the ability to detect, or it may not, depending upon the sensitivity of the test. All right, now let's say in the perfect world, if your immune system is functioning like it should be, and we get lucky as far as what the antibodies can recognize, when you get a second viral exposure, what's going to happen, and remember this is a log scale, so we're going up tenfold here, you get a massive production of IgG. That IgG, very quickly, that antibody neutralizes that virus, and you may not even get sick, you may not have symptoms. Either way, you recover quickly. So this is the ideal situation. This is how vaccines work, and this is what we're hoping happens if you've had it once and that you can't get it again. All right, let's get on to the test. There are three categories of tests that are out there today. The first is called the rapid diagnostic test. You may have seen images of these. They're really straightforward. You've got a little well right here. You put a drop of blood in here. The blood migrates up and interacts with some reagents. And then you have these three lines, potential lines. The first one, C, stands for control. You need to always see a red there. G stands for IgG. M stands for IgM. So remember, these are the two types of antibodies. And if you have a line for all three, that means that you have antibodies, potentially, most likely, you have antibodies that are recognizing COVID-19. Now, the benefit of the rapid diagnostic test is, first of all, it is fast and easy to use, right? You prick a finger, you put a drop of blood down, 15 minutes later, and sometimes even faster, you have results. They are cheap. I looked this morning, these things are $8 a piece. If you're buying them wholesale, I don't know what they get charged in clinical settings, but $8 if you buy them from the manufacturer directly. And if you can get a lot of them, you can do high throughput. That's the advantage of these. The downside is sensitivity. So if you think back to that image I just showed you of antibody concentration, what's called antibody titer, that appears in somebody's serum. The question is, how low can these guys detect? So in other words, how much antibody do you have to be detectable to get a positive? What they don't tell you also is concentration. So remember I said that some of us are going to have a much more robust antibody response than others. And so this test just tells you, do you have antibodies or do you not? And are they detectable? But it doesn't tell you if you have a lot of them or a little of them. And that actually is important to know. And then last and most importantly, this test, as well as all of these tests, is only as good as the specificity. So how good is the molecular machinery of this test in actually finding those antibodies that we think can recognize COVID-19? So let's look under the hood, if you will, and let's examine how this test actually works. So this is, if we were to look under the hood, this is the well where you're going to put the blood, right? And then immediately downstream from the blood's going to migrate this way, is you're going to have a little trap here. And this trap, actually, I shouldn't say trap, it's really another well. And that well is going to have some part of the COVID-19 virus, most likely some piece of the spike protein, since that's on the outside, or maybe the entire spike protein, but probably just a piece. And it's been manipulated in such a way that it will now react. So sometimes they add a gold to it. Sometimes it can be something that fluoresces. Sometimes it's just an enzyme that will change colors, but it's going to do something that's visual, right? After your blood and your antibodies migrates through here, it's going to keep migrating down the slide until it gets to a trap. And so how do you trap an antibody? Well, you trap an antibody with an antibody. So the trap is filled with antibodies that bind to your IgM. So you're going to be trying to trap IgM here that has potentially picked up this COVID-19 spike protein and then see if you can detect it. The blood sample will continue to travel down till you get to the second trap. And the second trap is, once again, an antibody that traps an antibody. So this antibody is going to trap IgG, right? So this is where your IgG line will show up. This is where your IgM line will show up. Now, I'm not showing the control, but it is here. All right, so let's actually walk through how this might work. So you prick your finger, put a drop of blood down. Remember, blood is where you're going to find, in your serum is where you're going to find your antibodies. Those antibodies are then going to migrate into that second well where you're going to have these COVID-19 spike proteins. They have some sort of marker on them so you can see them. And if you have antibodies that recognize that protein, they will bind, they'll migrate down, and then when they come to the first trap, IgM will get trapped, and that will make that little red line that we saw. IgG will keep migrating down, it will get trapped, and if it was able to bind COVID-19, once again, you'll get that line that shows up there. So essentially what happens is when you put your blood here, migrates down. If you trap something in this trap right here that contain the COVID-19, you'll get a red line. If you trap something in this second trap, you get this red line. Okay, so they're fast, they're cheap, perhaps not that sensitive, and they don't tell you anything about concentration of antibody. Right, the second type of assay is called an ELISA assay, which stands for enzyme-linked immunosorbent assay. Of course, that is why everyone says ELISA, because saying enzyme-linked immunosorbent assay is just a mouthful. I will say that ELISA did win the Nobel Prize sometime, I think in the 90s, and it is a very powerful technique. It is unbelievably sensitive, so you can pick up very small amounts of what you're looking for. In this case, we're looking for antibodies. And it can actually determine antibody levels. So it can tell you how much of an antibody you have. So it can give you titer/concentration. course, there must be a downside. The downside, it is slower to do because it's more sensitive. It is more costly. And once again, as always, the test is only good as the specificity. Can you actually find what you're looking for? Right, so how do these things work? So ELISAs are done in what are called microtiter plates. So they look like this. They're these little plastic plates that have these wells in them. They have 96, so you can do 96 samples at once. If we were just to look inside of one of those wells, so if we zoom in on one of those wells, what we would see is some part of COVID-19, probably the spike protein once again, since that's on the outside. And then the idea is you're going to take patient serum. Remember, this is where the antibodies are. You're going to add it to that well. You're going to ask the question, do any of these antibodies bind to this COVID-19 protein, spike protein? Then, and here's the brilliant part, you add a second antibody. Remember, how do you trap an antibody? With an antibody. You add this second antibody that binds to the first one. The second one has something on it that we can actually visualize. So it's going to amplify the signal and it'll give us a way to actually see it. And in this case, what it does is it's going to change it, in my example, yellow. Right, so what do the results actually look like? So this is a microtiter plate. Each one of these contains sera from a different individual. Down here, the ones that are clear, this bottom left corner, these 4 individuals did not have antibodies to COVID-19. Up here in the top right, notice how it's faint yellow. This individual does have antibodies to COVID-19, which I show here through binding, but notice it's not very bright yellow, which means there's not a lot of those antibodies. Compare that to this nice bright yellow spot here. So this individual also has antibodies that bind to COVID-19. And as I'm showing here in my little figure, there's a lot more of them. So it's more sensitive and it can give you an idea of quantity, like the titer, how much antibody does an individual actually have? All right, the gold standard, probably the best way to measure antibody success is called a neutralization assay. It is the most clinically relevant. It is highly sensitive. It can also tell you antibody levels. Why isn't it used like widely? It's really expensive. I shouldn't say really expensive. It's the most expensive of the assays, and it is the slowest of the assays. Whereas the first one I was telling you about, prick the finger, put it on the slide, results in 15 minutes. This is maybe 2 to 4 days before you get results. Let's look under the hood at how it works. So essentially you're gonna have a test tube in this case, and you're gonna have a patient's serum in here. Now remember, the serum is where the antibodies are. You're gonna take COVID-19 live virus and put it in there. All right, so you're gonna let the two of them interact. And essentially you're asking the question, does this individual have antibodies that can bind to COVID-19? Then you're gonna take this whole tube, ideally with COVID-19, with all these antibodies bound to it, and you're gonna dump it onto cells. These are probably human cells or a monkey cell line, but something that COVID-19 can infect. So you dump it onto living cells, you wait about 48 hours, and then you ask the question, was COVID-19 able to infect and kill these cells? And if it can kill and infect them, you get what are called plaques, these little zones of death, which you can count. And you can determine exactly how effective your antibodies were at neutralizing the virus. And so you can see why it's the most clinically relevant, because you're not asking how many antibodies you have. We are asking that, but you're also asking how effective are these antibodies. Okay, let me stop sharing for a second so I can see everybody. So that's a brief primer on how those assays work, and a very, very brief tutorial on antibodies for those immunology friends out there. I know I left out a lot of the details, but that wasn't my goal to go deep. My name is Mark Lubkowitz, St. Michael's College, rising from the ashes like the phoenix, trying to let crisis bring out the best of me. I'll see you soon. I don't know what's next on the docket.